The final is cumulative in this class.
Oh, good. Thank you.
Yes. And the final in this class this semester, if you've heard anything about previous semesters, this semester the final is going to be partly department generated. So it's not just me making the final exam.
Thank you.
Yeah. You can thank all of the other instructors who aren't doing their job.
Oh.
Calling them out.
What are their names?
I won't name names.
There's
but
one in any job you ever have.
there is a whole list, unfortunately, around here. So tell us how you do it. Right? Every time I look, what is going on with these people? All right, so let's get back to it. All right. We're really going to start connecting the dots with metabolism. We're doing like 4 or 5 different exercises in the lab that are all coming back to this. So we're going to do stuff regulating temperature. We've got a lab that regulates pH. And you'll see how you change those characteristics, how it alters the growth of an organism. We were talking about enzymes last time, and we were saying basically those enzymes are the key to metabolism, the key to making all of this work. Because the enzymes are lowering that activation energy. So they speed up chemical reactions by lowering the amount of energy it takes to start that reaction. Things that affect enzymes include temperature, pH, salt concentration, all of those components. We talked about that kind of stuff in chapter 4 and what the importance of it was. And we said it was important to understand how much salt you were putting in with an organism in order to regulate growth. It's important to understand that pH regulates growth of an organism, and without being in the right pH level, the organism isn't going to grow appropriately. All of those conditions are coming back to altering the enzymes of the organism and to controlling its metabolism. When it comes to these enzymes, enzymes are literally the key to making all of this work inside an organism. So your enzymes have to be balanced and they have to work appropriately, just like these microbial enzymes have to work appropriately. When we talked about taking a multivitamin, and we said, look at all of the different nutrients that are in there, the different components, the different minerals, all of the different components in that are ultimately coming back to making sure that your enzymes are functioning appropriately. When your enzymes don't work right, you don't feel right. You're not at your best. Same thing happens with microorganisms. Microorganisms can actually regulate their enzymes. Your bodies do this as well. So an organism needs an enzyme, but maybe it only needs an enzyme when a certain nutrient is present. Maybe it doesn't need it all the time. There's no point in continuously making an enzyme if the chemical reaction it facilitates is not needed at that particular point in time, okay? So there's, this is like, cleaning out the clutter in your house, okay? If you don't need it or you're not wearing it, get rid of it. The same thing happens with these microbes. They look at an enzyme and they say, okay, this is made of protein, so it's got amino acids, it's got a carbon backbone. If we don't need that enzyme at that particular point in time, degrade it, get rid of it, use it to make something else. So in regulating enzymes, there are 2 main types of regulation. Both of these are shutting down an enzyme. Both of these are known as inhibition, which you see written at the top. So we have competitive inhibition and we have non-competitive inhibition. The way these 2 are functioning is all coming back to what was called that active site. So if you remember, we talked about the active site as being the site on the enzyme where the substrate binds to it and then acts on it, okay? In competitive inhibition, your normal substrate and your competitor are both, or your inhibitor, are both competing for that active site. When the normal substrate binds to the enzyme, the reaction proceeds. When the inhibitor binds to the enzyme, the reaction is actually blocked. Lots and lots and lots of different types of antibiotics and other drugs work through this exact process. They are competing for an active site on an enzyme. In a few weeks, we're going to get into antibiotics and how they work. And we'll talk about the fact that when you get an antibiotic, they tell you to take it twice a day or take it 3 times a day. They tell you to take it twice a day. What does that mean? How often are you taking it?
Every 12 hours.
Every 12 hours. If they tell you 3 times a day, it's every 8 hours. It doesn't mean when you feel like it, as long as you take 2 pills or 3 pills a day. It means you've got to have the timing down. The reason the timing has to be right is because you have to drive this process here. The drug has to be of the right concentration in your body to shut down the enzyme and to shut down bacterial growth. When the drug is not at the right concentration in your body, the enzyme is still functioning, the microbe is still growing, and your infection is not getting any better. So patients who refuse to take their medication at the right time should drive you absolutely nuts. Because they do, because of this process, because they don't understand that it's all about the timing. If you want that drug to work, the only way it does its job is if the concentration is right in your body continuously. So not taking it at the right time drives this process, when in fact taking that antibiotic, you want it to shut the organism down, to shut the enzymes down, which kills the organism because it's shutting down the growth. There is another form of inhibition known as non-competitive inhibition. In this case, you're not competing for that active site. What happens in non-competitive inhibition is that a regulatory molecule, which is normally a product way down the line, signals back to the enzyme and tells it to shut off or shut down. You can think of this as almost a feedback loop, okay? The product at the end says, wait a minute, you're making too much of this. I've got enough here. We're good to go. You don't need to drive enzyme production way early on. So that product binds to an early enzyme and shuts down the reaction, turning it off, okay? Competitive inhibition, I look at this like you're competing for the same thing, okay? You're fighting to get the last cookie. You're fighting, think of the enzyme as being a lock and key, okay? When I was young, I would always like fight with my brother to see who could get to the door faster when we got off school, okay? It was who got their key in the door faster, who got in the door faster, because then you got to choose what you wanted to watch on TV, okay? We were fighting for the same thing, all right? And if I got in first and he was being a slow poker making me angry that day, I knew I could shut the door and I knew I could stand there with my hand on the deadbolt. Your hand is on the deadbolt. Doesn't matter that that key goes in there, it's not doing anything, okay? So non-competitive inhibition is shutting something down by a different site. If you think of the enzyme like a lock and key, your key has to go in. So the substrate has to go into the active site in order for the enzyme to work. If something else is blocking that active site, the enzyme doesn't work, the lock doesn't work. If you've got someone holding down the back, it doesn't matter that the key is there, the lock doesn't work, okay? So it's ways of shutting enzymes down, 2 totally different pathways. One is competing for that active site, concentration is important. And the other one is saying, I don't care what's going on right now, the product in the end is shutting you down, okay?
So it's essentially like changing the shape?
It changes the shape. The non-competitive inhibition changes the shape of that active site so it doesn't bind. Why would you want this? When would you want this? Your body does this all the time. You make an enzyme known as lactase. Lactase is an important enzyme because it breaks down lactose. I see some yogurt sitting here, okay? Someone had string cheese earlier, okay? If you're lactose intolerant, okay? We're gonna get to that one in just a second. So if you are eating lactose, dairy products, your body goes, oh, I need to make the lactase enzyme so that I can break down the lactose and I can get the energy from it. When you don't have dairy, your body shuts down that pathway. Don't make the lactase enzyme if you don't need it. If you're lactose intolerant, what makes you lactose intolerant is that your body is no longer producing the lactase enzyme. Newsflash, it naturally happens as you get older, your body loses the ability to make lactase. Okay? There can be genetic factors that factor into that as well, but your body naturally loses that ability. So if you are lactose intolerant, you can still have lactose. You just typically take what's labeled a dairy aid or lactate or something like that along with it. It's a pill that you can swallow when you have dairy. That pill is powdered enzyme. That's all it is. You take a set amount of pills depending on how much dairy you eat. That controls the signs and symptoms that you have of being lactose intolerant. If you're not having dairy, you don't need to have those pills because you don't need any of that enzyme. All right? Yeah, Rachel.
So for like cystic fibrosis, isn't that like where you're missing enzymes like in your pancreas that like help break up mucus?
It's a genetic issue. It's not. It's typically not
related?
enzyme related.
I couldn't remember.
Yeah.
It's like a build-up.
Yeah.
Yeah, I just remember that your pancreas doesn't like signal like the like.
It's about the
something to break up.
mucociliary
Yeah.
mechanisms that
Yeah.
help break it up and move it out.
Yeah, I just couldn't
Do
remember
not
if those
believe it's
mechanisms were
specifically
enzymes
enzyme
or not.
related. Yeah. Here's a way of looking at a feedback loop. Okay. So you're making an end product. Starting compound, end product. You have intermediates. Remember, these are also referred to. Okay. In the long run, okay, metabolic components. Each enzyme is involved in one chemical reaction. If you've got too much end product, you don't just shut down the last chemical reaction in the pathway. Because then you're going to create too much of this when you don't need it either. If you're making too much end product, it shuts down the first enzyme in the pathway to shut down this entire biochemical pathway, okay? So regulating enzymes is really important in regulating the overall growth of the organism. You can regulate enzymes by temperature, pH, salt concentration. You can also regulate them by having inhibitors present, either competitive inhibitors or non-competitive inhibitors. Enzymes in disease is a big deal. You want to know when you get an infection, what organism is causing your infection, and you want to be able to understand what it's releasing. Okay? So lots of pathogens actually secrete exoenzymes. Remember, exoenzymes exit the cell. So we talked about cholera before. How that organism, Vibrio cholera, causes vomiting, diarrhea. You lose gallons of water a day. What's actually happening is that the Vibrio cholera, when it gets in, it colonizes your gut. And it's actually really cool and highly functioning smart organism because it multiplies first and then it goes, okay, now we've got an army, now we're all going to release the exoenzyme at once. So Vibrio cholera, that disease comes on really suddenly because all of the bacteria release the exoenzyme all at once. That exoenzyme causes your cells to then lose all of their water and start leaching water, okay? All it is an enzyme the organism is releasing that causes all of those downstream symptoms in your body. Similar things happen with diphtheria, botulism as well. The signs and symptoms of botulism, so really extreme food poisoning, can lead to death, bad stuff. The causative agent of all of those signs and symptoms is actually an enzyme that the organism releases. And it basically is a neurotoxin in your system that destroys your nerves and leads to all kinds of other things. When you get, if you get, Botox, okay? Botox is literally the botulinum toxin. It's an enzyme. They've taken that enzyme, they modify it a little bit, and that's what they inject to get rid of your laugh lines and other stuff. The enzyme itself is a neurotoxin in your system. Okay?
We were talking about laugh lines before lecture started.
Very nice. Uh-oh.
I love how you look at her.
She's laughing. It's all good.
Just one in the group.
See?
So you're paying $250 for enzymes?
Yeah.
Worth it.
You're just paying lots of money for enzymes. That's it. Okay? The enzymes are important to the organism. When an organism causes an infection, it doesn't care if it makes you sick. That's not its goal. Its goal is to get into your body and reproduce. That's its goal. It just wants to make more of itself. These enzymes typically are helping it get around your host defenses, move around in your body. If they happen to create signs and symptoms and disease along the way, then too bad, so sad for you. The organism doesn't care. Its goal is to reproduce. those enzymes help it avoid your immune system, avoid other reactions in your body which are trying to fight it off, and allow it to stick around longer, which means it multiplies more. Okay? The organisms can increase the ability to cause disease, keep the cells alive. So there's a lot of reasons why the organism would make those enzymes. They just happen to be poisonous to you in the long run. Okay? So enzymes are really big in terms of disease. They're also really big in terms of the overall growth of the organism. So let's fill in the blanks here. Metabolism uses what to catalyze reactions?
Enzymes?
Enzymes. If they break down, what is that called? Catabolism. So that break down or do catabolism, catabolize organic molecules, ooh, I spelled that wrong, into intermediates, okay? Intermediate molecules, and also uses enzymes to use those precursors to build. What is building?
That's the anabolic.
Anabolic. So anabolism, anabolic, to build larger, more complex molecules that are particularly suited to them. What's involved in a biochemical pathway? What do we need to start with? Our starting compound is also called what? Substrate. We make intermediates, right? And then we make the products. Okay, so we're dealing with biochemical pathways. What is a pathway? I'm adding more to it. What's a pathway? Now write down. What is a pathway?
Yeah, but where's substrate going?
We're talking through it.
Okay.
What's a biochemical pathway? A series of what?
Reaction.
Chemical reactions. Okay, so a pathway is simply a series of chemical reactions which take substrates to products.
Says that cystic fibrosis clogs the pancreas and digestive enzymes are not able to get to the intestine.
So it doesn't alter the
doesn't-
enzyme.
No,
it
just,
doesn't
like,
change the
but it
enzymes.
affects
Right.
some of them.
It affects
I was
their
like,
ability to
I
work.
remember
in the
reading
end.
something about it.
They're still produced.
Yeah.
All right, so energy is needed in large quantities for what parts of metabolism?
For the breakdown?
The anabolic. So needed in large quantities for the anabolic parts of metabolism. Energy is produced during the catabolic parts.
So you said pathways of series of chemical reactions that take substrates from products.
To take right. A pathway is a series
Substrate.
of chemical reactions that take substrates to products.
The products to the subjects to the product, substrates to the products.
Yeah, I work on my face. Leave me alone.
Keep the romance is ending.
Right? It's getting real around here. Okay. No, yeah, there will be no Valentine's Day gifts exchanged down there. All right.
He did buy me Skittles.
Oh.
It's serious.
See?
It's more than I'm getting, so.
Yikes.
Well, this is getting depressing.
All right. We're digressing here, seriously. All right, so remember, all organisms need a carbon source and an energy source. The carbon source is used because they need carbon to do anabolism. The energy source is used because we have to make ATP, which is then also used in anabolism. So all organisms need a source of energy, which is basically just the compound broken down to make ATP. Harvesting that energy is a series of coupled reactions, which you have heard of before, known as oxidation reduction reactions. And you're probably like, I know I've heard the term, but I don't remember why it's important or what it actually does, okay? So what happens in these reactions is it's all about transferring electrons. You remember back to your chemistry, the electrons are negatively charged particles that move around the atom in these orbitals around the outside of the nucleus. All organisms, all atoms have these electrons, okay? What's happening in oxidation reduction reactions is simply just a transfer of electrons. You are playing hot potato, and the potato is the electron. That's it, okay? So when a compound, so in this case, compound A has the electron, and it's transferring the electron to compound B. So compound A loses the electron, it becomes oxidized. Compound B gains the electron, and it becomes reduced. This is totally opposite. You would think reduced means I lose it. It's not the way it works with the electron. Keep in mind these electrons are in those electron shells outside of the nucleus. They're negatively charged. How they are rotating and where the mass is in terms of the orbitals is what's actually coming back here. So if we gain an electron, we're considered to be reduced. If we lose an electron, we are oxidized. Tossing around the electrons ultimately allows a cell to harvest the energy from those reactions and to use it to make ATP. So transferring electrons from one compound to the next, you do that in chemical reactions, and those chemical reactions are going to allow us to make ATP. We talked about this before when we talked about phosphorylation and making ATP, and we said there were two different types of phosphorylation. Substrate level phosphorylation, okay, and direct phosphorylation, so we're directly making ATP versus indirectly making ATP. The indirect form is coming in the form of moving around those electrons. And that indirect form is going to be, we're going to talk about that in terms of the electron transport chain in a little bit.
So the N5 for compound A, when it's oxidized, it would be a donor, and then for compound B, it would be a carrier.
No. Those terms
Because
don't
it
count.
gains. I'm sorry, I remember.
You're trying. You're trying to remember your chemistry. If you, I wouldn't think so, donor versus acceptor.
Yeah.
You can use those terms, but ultimately it's coming back to oxidation versus reduction. So we're always going to refer to them here in terms of which one is oxidized and which one is reduced. So if you think, okay, the donor becomes oxidized, the acceptor is becoming reduced, then you're moving along, okay? Yeah. And really, this is going to keep going. This is going to keep transferring it back and forth and back and forth and back and forth. It's ultimately going to be who gets the electron in the end. So think of it just like a game of hot potato where you're constantly transferring the electron from one to the next to the next. It's ultimately about I may become reduced, but then I become oxidized because I pass it on to the next one. So that constant exchange of electrons is what's going to allow you to make ATP.
When I think oxidation, I think rusting. Is that right? That's what kind of.
Yeah, because
Right.
it's oxidizing.
Right. As it oxidizes, it rusts. It's ultimately coming back to
Making
the
sure that's
breakdown
still
of the
right in
compound. Yeah. All right.
Definitely like.
You may have heard of these things called electron carriers. Okay? NAD, FAD, and NADP. All these electron carriers are little taxis that are functioning in your hot potato of electrons, moving things from one place to the next. Okay? So the oxidized form is going to have this positive at the end. What do you think that positive is referring to?
That it lost the charge because it gave it.
Charge, okay? It's positively charged. When it gains the electrons, then it becomes reduced. When it gains the electrons, now we're adding on, in this case you see a hydrogen. Hydrogen is atomic number what?
2. 1.
1.
Shoot.
Which means it has how many protons?
One.
1. How many neutrons? How many electrons? Well, it depends on ions or not. How many electrons does hydrogen have? 1. Okay? So when you start moving around, hydrogen ions, if it's got just a positive charge, this means it's just a proton. Okay? Remember, the protons are positively charged, the electrons are negatively charged. If you just see this H, electrically, this is neutral, neutrally charged. It means it's got one proton and one electron. If you see H plus, the way it's got this positive charge is because it's simply just one proton. This one proton, one electron. Okay? If you add on an electron, negatively charged, there's no way to eliminate the proton in here. If you remove the proton, it's no longer hydrogen. Okay? So I'm not going to draw an H negative with just an electron because it's not hydrogen anymore. Okay? E negative means you've got one electron. Okay? So what's happening here is that you're going from this positively charged electron carrier. In each case, it's accepting 2 electrons, and it's taking 2 hydrogen ions. And it's giving you these reduced forms of NADH, FADH2, and NADPH. All it is a carrier of electrons. It's helping to move the electron from one spot to the next. It's a little taxi for electrons. When it's Uber, there we go, it's Uber for your electrons, okay? Taxi doesn't apply.
Living in the 90s here.
Now, yep, right now it's showing how old I am, okay? So if the H is on the end, that means it's in its reduced form. If you don't see an H and you just see the positive, that means it's in its oxidized form and it's waiting to get its electrons, okay? Why do we need NADH, FADH2, and NADPH? They're ultimately going to help make ATP synthesis and drive biosynthesis of compounds, okay? In its reduced form, it represents what's called reducing power. Reducing power means we can make ATP. Reducing power equals ability to make ATP. Because it's got usable energy in the bonds, okay? So if it's got the H on the end, the Uber driver has picked up its electrons.
Uber.
Oh my gosh. And it's getting ready, it's going to use that to make ATP.
Not Uber driver.
What?
Uber. It's
Uber?
Yeah, Uber. Trust me.
Oh my lordy. Okay.
Like, I'm adding an extra glass of wine tonight.
Yeah, right? I may be drinking earlier tonight.
What's in the thermos?
I'm
Right?
glad I closed that.
All right. Don't focus on memorizing the slide, okay? There's a lot going on here. The point here is that all of these arrows are showing you chemical reactions, okay? And we're looking at different types of pathways. Look under glycolysis. This is that branched pathway we talked about. Here, with TCA cycle, this is that cyclical pathway we talked about. All of these are different pathways, ultimately for the organism, okay, that an organism can use. And what you see is we're showing glycolysis, the TCA cycle, fermentation, respiration, which means we're using an electron transport chain. Ultimately, the goal here is to make ATP. ATP. There's some ATP up there. Where you see this guy reducing power in this awesome little box. Reducing power means the ability to make more ATP. Okay? All of your reducing power is going to get funneled into either respiration or fermentation, into the electron transport chain. So if you see respiration on the end, it means it's got an electron transport chain. So we make directly ATP, but we take all of these guys, these little Ubers, and we put them all together to make more ATP in the end. It's a way of being efficient with the cell's energy source, okay? All of these are catabolic pathways. Catabolic is doing what? Breaking down. So if we are breaking things down, we're going to produce energy, which means we're making what? ATP. We're also going to generate reducing power, which is NAD. NADH, FADH2, okay? And we're also going to make those precursor molecules, which are also called intermediates. They mean the same thing, okay? A goes to B, goes to C, goes to D. B and C are your intermediates, or your precursor molecules, okay? Yikes. I love this table, okay? This is like, this is the terms from Anchorman the other day. Did anyone see that? Like, I love lamp, okay? I love this table, all right?
I love
This
it.
table, right? This table summarizes everything we're gonna go through in the next rest of the slides for this particular chapter,
So
okay?
memorize this.
If you memorize this, you would be in good shape, but you have to be able to walk your way through it.
Wow.
Okay.
Well, I'm good at memorizing
You're
part.
good at the
We
memorizing
did
part.
this
We'll
in
work on the
bio.
walking our way through it.
Don't know
This
how to
whole thing
walk,
in
so.
bio, like Gen
See?
bio. Yeah.
Yeah. This is why it's a prereq.
Yeah, I'm like,
I'm
I
good at
did it.
memorizing, but I can't
Oh, I
walk.
remember.
so-
Alright, so you've got 3 different pathways we're gonna talk
it's
about.
easy. It's just memory.
Aerobic respiration, anaerobic respiration, and fermentation. If you see respiration written, it means it's got to have an electron transport chain. The next column over literally says uses an electron transport chain. Under aerobic and anaerobic, it says yes. And under fermentation, it says no. Okay? So if it says respiration, it means it has to do an electron transport chain. Has an electron transport chain.
I put train.
Train. A train works too. Just shuttling them around. It's fine.
It's the chain train.
It's the longest chemoorganohead.
Between the chain gang?
Oh,
Okay.
here we go.
What?
Now,
Stuff out of it.
remember what we're doing. We're passing around electrons. We're doing oxidation reduction reactions to utilize the energy from our energy source and to make ATP. So, ultimately with hot potato, somebody has to end up with the potato. It's got to go somewhere. It doesn't just like drop on the floor, okay?
Savage.
All of these have to have a terminal electron acceptor, and what that means is what compound ultimately takes that electron. If it's aerobic respiration, aerobic, your terminal electron acceptor is oxygen. It's not just any oxygen though, it's molecular oxygen, it's in the form of O2. Yes, this comes back to your chemistry and back to those electron shells and the fact that you're never going to find an oxygen atom hanging out by itself because it's unstable with its electron shells. It's going to be in the form of O2.
It's gonna be fine.
It's going to be fine.
I'll agree, okay.
In the event it's anaerobic respiration, you can use something other than molecular oxygen, okay? Now, I am never going to give you a chemical reaction and say which one of these would be the terminal electron acceptor if we do anaerobic respiration.
You never asked that? Or
No.
you would ask?
I'm not going to have you distinguish, does it need nitrate, does it need nitrite, does it need sulfate, what's going on, which one does it need, and when does it need it, okay? It doesn't matter. I don't think it matters, okay? If it's doing anaerobic respiration, the final electron acceptor is always going to be something other than O2. You get all kinds of weird organisms that do anaerobic respiration that use some obscure compound as its terminal electron acceptor. For this, if you know it says anaerobic respiration, know it does have an electron transport chain, and know that the final electron acceptor is something other than molecular oxygen, okay? You're going to benefit here because I was traumatized in college, okay, by a class where I literally had to memorize all of the electron acceptors and how many electrons were produced and where the growth would be, and it was a whole bunch of just charts, and this makes ammonia, and that, and I hated it with a passion, okay?
But yet you love this.
It doesn't matter. I do like this. When I realized in the end it ultimately doesn't matter, then I was like, that's just a waste of my time.
Sounds like
So I wasn't happy about that, okay? If it does anaerobic respiration, its terminal electron acceptor is something other than molecular oxygen. If it does fermentation, it's using some other organic molecule, okay? Bottom line, if you hear aerobic respiration, it means oxygen is its final electron acceptor. If it doesn't say aerobic respiration, it's using something else as the final electron acceptor. The electrons have to go somewhere in the end. We doing okay?
Yep.
Yeah.
For the anaerobic,
Yeah.
is it always going to be like a compound like that?
Yes.
It's not going to be like carbon?
No, it's always going to
Okay.
be a compound, and what you notice is that these compounds are all negatively charged,
Okay.
which means they're looking for that electron.
Got it.
I want an electron to make myself more stable,
Mhm.
so that's typically where they're going. Next column, next set of columns. ATP generated by substrate level phosphorylation and ATP generated by oxidative phosphorylation. Substrate level phosphorylation is the direct production of ATP. I do this chemical reaction, ATP pops out. Oxidative phosphorylation is where we are doing oxidation reduction reactions. We're passing around the electron, and we ultimately are going to create ATP indirectly. Okay, so the direct production of ATP happens in glycolysis and the TCA cycle. So what you see here is an aerobic respiration. You get 2 from glycolysis, 2 from the TCA cycle, you get 4 total. That's it. It only makes 4 ATP from breaking down glucose in direct production. In anaerobic respiration, the number is going to vary because there's different carriers used. It's less than that of aerobic respiration, but more than that of fermentation. It's literally just in the middle. And it's going to be different for every organism depending on its enzymes and depending on what that terminal electron acceptor is. Fermentation only uses glycolysis, so your number here is simply 2. Okay? Oxidative phosphorylation is the next one. Oxidative phosphorylation, ATP is only generated in electron transport. So, aerobic respiration makes a total of 34 ATP in oxidative phosphorylation. In other words, the electron transport chain, or train, is really important for these organisms because that's where they're making their most ATP. In anaerobic respiration as well, they're going to make a decent amount of ATP in that electron transport chain. It is simply going to vary for every organism. Why is the number 0 here under fermentation?
There's no oxygen.
Because there's no electron transport chain.
Oh, yeah,
No electron transport chain means no oxidative phosphorylation. Adding up your numbers. 38 is your theoretical maximum in aerobic respiration. It says theoretical maximum because organisms could decide, wait a minute, I need that enzyme over here, or I need this compound over here, and they make less than that amount. But 4 plus 34 gives you 38. Okay? The most efficient way to break down carbohydrates and make the most ATP is through aerobic respiration. Anaerobic respiration is also going to do these products, but it's going to be less efficient. Typically, the theoretical maximum with anaerobic respiration is somewhere 34 or less. Could be 36, depends on the organism, but it's less efficient than aerobic. Fermentation only makes 2 ATP. What this means is that if a cell wants to make the most ATP possible, it would be much better off completing aerobic respiration. In the event oxygen is not around or it cannot use it, that is when it would use either anaerobic respiration or fermentation. But those 2 processes are less efficient. Depends on what compounds are available, and it depends on what enzymes an organism has.
Can it like go through aerobic and then continue on to do one of the other ones to gain more?
So it will do aerobic as long as it can and as long as there is oxygen present. It's going to start with sugar. If it runs out of oxygen and there's still sugar present, it depends on if the organism can utilize another enzyme or not. If it can, it's possible it can switch to a different pathway. But if it can't, there's nothing it
But
does.
it won't just like switch back and forth as it.
Most of them will not switch back and forth. It will pick the most efficient until it has no ability to do that anymore.
So is that similar to like when if we were like exercising and we reached like that anaerobic like threshold?
Right.
So we like run out.
You run out, right. So if you're exercising, if you're exercising, you get that muscle cramps and buildup of lactic acid in your muscle, like runners do and stuff like that. That happens because they basically deplete all the oxygen. You drive fermentation in there instead, and then you produce this lactic acid as a byproduct. Yeah.
So BCAAs are good for?
In the event, the number here, this is looking at a prokaryotic organism, and it's specific for chemoorganoheterotrophs. So this is telling you where we're utilizing and looking at prokaryotes. So think bacteria here, okay? And chemoorganoheterotroph, what do you remember about that term?
Chemical compounds.
Right.
Carbon.
So it's organic compounds, organic chemical compounds for both its carbon source and its energy source. So these are bacteria that are basically using sugar as an energy source. They're breaking down glucose, for example. In the event you look up some random video, okay, or you look into your basic biology textbook and you're looking at it in a eukaryote, these numbers are not the same in a eukaryote. Eukaryotes, metabolism is going to happen, or respiration is going to happen in a mitochondrion, okay? You lose ATP crossing along the membranes of the mitochondria, and that's why the numbers are actually going to reduce. Prokaryotes do not have mitochondria, so therefore you're not going to see that. So the numbers here are specific to bacteria. If you are asked a question on an exam, it's going to be specific to bacteria.
Love it.
Love it.
Mm-hmm.
It's all coming together. All we are talking about here are chemical pathways. And these are chemical pathways that are doing catabolism, so they're breaking things down. The whole point of that is we want to make ATP. If we directly make ATP, remember that's substrate level phosphorylation. If we generate reducing power equals the ability to make more ATP. That's oxidative phosphorylation. So our catabolic pathways are important because it does not only direct production of ATP through substrate level phosphorylation, but it also allows us to make more ATP later through oxidative phosphorylation. Remember, those chemical pathways are just chemical reactions lined up one after the next. So we have a starting material. That starting compound we turn into intermediates. We also make end products from that, okay? Anything in the middle is referred to as a precursor metabolite, which can be taken apart and then used in anabolic processes. Hey, you made this over here. I know you're not finished with it yet, but let me borrow it and put some of it over on this side. Let me take it and use it over here, okay? Anything generated inside the cell can be diverted from one spot and be used in all kinds of other ways. This is a figure from a different textbook, but I really like it because it gives you all three pathways next to one another and shows you where they're the same and where they're different, okay? Aerobic respiration, anaerobic respiration, fermentation. Every one of them starts with this process of glycolysis. It's taking glucose and turning it into pyruvate. Every one of them does that step. If you are doing aerobic or anaerobic respiration, it's going to then convert it through a transition step into acetyl-CoA, and that acetyl-CoA is then going to enter into the Krebs cycle or the TCA cycle. Krebs, TCA mean the same exact thing. TCA is citric acid cycle, okay? They're referring to the same exact thing. Depending on the book you pick up or the figure you see, some will say Krebs and some will say TCA. Same thing. The acetyl-CoA goes into this process and gets completely broken down. As part of that, we're making some ATP, but we're also making our lovely friends, the Uber drivers, NADH and FADH2, those reduced forms. Those forms bring electrons to the electron transport chain, which makes more ATP. In aerobic respiration, oxygen is your final electron acceptor to give you a total ATP production of 38. In anaerobic respiration, we're putting it on some non-oxygen acceptor. Our number is going to be inefficient compared, somewhat more inefficient, less efficient, more inefficient to aerobic respiration. Typically the number is somewhere between 2 and 36. Doesn't really matter. Just know it's less efficient. Fermentation is totally different because once you finish with glycolysis, it takes it down the fermentation pathway to either make acidic byproducts or alcohol products. It's going to take that and use the final electron acceptor is going to be some organic molecule. But we're limited in terms of the amount of ATP produced. All three of these pathways start out the same. Fermentation breaks off first because the minute glycolysis is finished, it goes through fermentation instead to do something else with your pyruvate. Aerobic and anaerobic respiration continue through the transition step, through the TCA cycle, through to electron transport. Where these two differ is ultimately in the final electron acceptor. How are we doing?
Beautiful.
Awesome. There's 4 steps. You want to understand the 4 steps. You want to be able to walk through the 4 steps. So, we start always with glycolysis if we're doing a form of respiration. Then we have the transition step, sometimes referred to as pyruvate oxidation. Then you go into the TCA cycle, and then you go into the electron transport chain. We're going to talk about all 4 of these. Ultimately, you need to understand the starting materials, the end products. So what do we start with? What are we ultimately making in here? There are lots of individual chemical reactions in these processes. It is not about memorizing every chemical reaction. You don't have to do it. But you need to be able to put these 4 in order, and you need to be able to say what's my starting material and what's my end product. And I said that because this was the next slide, and I didn't want you to freak out with all the chemical reactions on here, okay? What's our starting material here?
Glucose.
Glucose, okay? So we're starting with glucose. Now, in order to work through this process, you actually need to break down 2 ATP in order to get this started. So you have to have an input of 2 ATP, otherwise this process never gets completed. What that means is if a cell loses all of its ATP, it's totally gone, it cannot start making more. So the goal is you never want to completely run out, because if you completely run out, you're not going to be able to make more in the end. Okay? You're not going to be able to get back on your feet. Okay? So we're starting with glucose. We're also going to require 2 ATP. Our products. Let's look at what we're making here. We're generating reducing power. Remember that reducing power is in the form of NADH. What you can see here is that the blue are going to be the carbon atoms. The yellow are actually the phosphates. So in breaking down the 2 ATP, you're actually putting 2 phosphates onto this molecule. We then are breaking it apart, and then we're simply doing a series of rearrangements to end up with our final end products. Okay? So in this process, we're making reducing power in the form of NADH. But how much NADH are we making? One, two, okay? We are generating ATP. ADP, making ATP here, so this is 1, this is 2. But look, we're making more down here, 3 and 4. So we actually make 4 ATP. And then our final end product in this biochemical pathway is this molecule of pyruvate. How many pyruvates do we have? 2. Okay? It's a 3-carbon sugar. So we started here with glucose. Glucose has how many carbons? 6. The chemical formula for glucose is C6H12O6. We've started with a compound that has 6 carbons. We're left with 2 3-carbon compounds. How many carbons do we still have?
1.
6.
There we go.
1, 2, 3, 4, 5, 6.
Oh, sorry.
So we still have the same amount of carbons. All we've done is basically broken this molecule apart. But in doing that, we've generated reducing power and we've generated ATP. So the point of this process, it starts the breakdown of glucose. Ultimately, how much ATP did we make here?
You didn't
Net?
make any.
Net.
2.
2. Okay? We made 4, but we had to put in 2. So we're only actually netting a gain of 2 ATP.
So then how do you ever gain more?
It goes through 4 steps.
So substrate,
Feels so
substrate production is 2 ATP. Look back here. Doot. Okay. ATP generated by substrate level phosphorylation. 2 in glycolysis. Net. 2 net ATP. Okay? We're laughing because we've had enough.
It's fine.
I just
It's
think how
fine.
this is like a shock for everyone. We went over all of this in bio. Now you know my frustration.
What do you know?
The next part. So this is step number 1.
I took it last lesson.
And I've run out of board.
We're
Okay.
on the board. You could just flip that up and
Right.
still project.
Yes, I think we're going to end up doing that.
Yeah.
So step number 2 is known as the transition step. It's also called pyruvate oxidation. If we oxidize it, what's happening to our electrons? Are we gaining them or losing them?
Losing.
We're losing them, okay? So pyruvate oxidation, we're losing electrons, we're breaking it down. So what happens here is that this is going to link glycolysis to the TCA cycle. But the whole point of this is that we take that 3-carbon sugar, which is pyruvate. This figure says pyruvic acid. It is the same exact thing. I know. Now we're getting into like multiple names for the same term. Okay. So it takes the pyruvate and it converts it into acetyl-CoA. Here we have, bless you, we have 3 carbons. When we're done, we have 2 carbons. So we're breaking this down. In the end, the carbon goes into making carbon dioxide. It gives off carbon dioxide. Byproduct. It's also, because we're giving off that carbon and we're oxidizing, we're gaining reducing power. So we're going to go from NAD to NADH. The coenzyme A is going to come in and it's going to make acetyl-CoA. Now, how many pyruvates did we end with at the end of glycolysis?
2.
2. Each one of those goes through this chemical reaction. So this thing happens twice for the one glucose that we started with. So, step 2, transition step. What are we starting with?
Pyruvate.
Starting with pyruvate, but how many?
2.
Our products.
Carbon dioxide.
Carbon dioxide, how many?
2.
2.
What?
I know, right? It's got to happen twice.
Oh, yeah.
Once, twice. We are making NADH. How many?
2.
2. We are making acetyl-CoA. How many?
2.
Okay. We went from, so here's another way of looking at it. We started with 6 carbons in that glucose molecule. How many carbons are we left with as part of the molecule at the end here of the transition step?
4. Total.
Total.
4.
4.
Ah.
Okay? 4. Because we've got 2 from this molecule, we've got 2 from the second molecule. Which is, we've lost, it all has to add up. So,
How do you see that?
pyruvate has 3 carbons, right?
Overall, this in bio.
Yeah, 3 carbons.
We lose a carbon,
Mm-hmm.
and we form acetyl-CoA, which has 2 carbons.
You're.
But the other carbon is in the form of CO2. So if we add this up, we have 6 carbons up here. Okay? We have 2 CO2, that means there's 2 carbons here. We have 2 acetyl-CoA. 2 plus another 2 is 4. 4 plus 2 gives us 6.
12. 6 plus 6 equals 12. 2 pyruvates.
Play this side.
If it's no, yeah. You're just making sense out of it.
This is how I make sense.
What are we starting with in the TCA cycle then?
2 carbon dioxide, 2 NADH.
ATP.
Acetyl-CoA. How many of those? 2. Okay. Our CO2 is literally just given off as a byproduct. You know when we talked before about how all those organisms give off gas? We said if you squish the milk carton and you come back later and it's bubbling on you, it's giving off gas. It's carbon dioxide. It's from this process here. All right, this diagram has the transition step at the top so that you can see it. The acetyl-CoA goes into this. This is a cyclical pathway now, so we're regenerating materials. Immediately, that CoA goes back up and gets recycled. The 2-carbon acetyl-CoA molecule, the 2 carbons from there join with a molecule of oxaloacetate. 2 plus 4 gives us a 6-carbon compound. Okay? The whole point of the TCA cycle is to complete the oxidation of glucose. The whole point of this is that we are exhausting and breaking apart every part of that original 6-carbon sugar that we started with. So this happens through a series of reactions in this cyclical pathway. We're going to make a little reducing power and we break off a carbon. Carbon always leaves in the form of CO2. We're going to do another rearrangement. We're going to make a little bit more reducing power. And we're also breaking off another carbon in the form of CO2. 2 carbons went in, 2 carbons have popped out. Our glucose molecule is gone by the time all of the acetyl-CoA goes through this process. The rest of this other half of this cyclical pathway is all about regenerating the starting material. When we do that, we're going to generate a little bit of ATP. We're going to generate more reducing power in the form of an FADH2 and in the form of an NADH molecule. It's got to happen how many times for the glucose molecule that we started with? 2. Okay? So, if we started, too short for this. If we started with 2 acetyl-CoA, we're now producing how many carbon dioxide? 1, 2, but we're doing it again. So, 4. We're also making reducing power, which now I've written all over.
FAD?
NADH.
Oh.
We're making 1, 2, 3, 6.
Yeah.
And we're going to make FADH2. But how many? You just have 1. 2.
So,
We're also making ATP. 1.
2?
2 total. The purpose here. So if we're here, our purpose is to start the breakdown of glucose. Here, our purpose is to make acetyl-CoA. Oh. The transition step is to transition the products from glycolysis so they can enter into the TCA cycle. The purpose of the TCA cycle is to complete the oxidation, which is actually, aka, breakdown of glucose.
So, how many times must that cycle happen for every glucose molecule you started with?
How many?
2?
Twice.
Ooh.
So glycolysis happens once. It's a branching pathway. The transition step is a linear pathway. It's got to happen once for every pyruvate that you start with. So it's got to happen twice. The TCA cycle is a cyclical pathway. It's got to happen once for every acetyl-CoA, which means it has to happen twice for every glucose.
Sorry, your work is beautiful.
No, I like, nope. You're good. How are we doing? We've covered 3 of the 4 processes.
There's another.
Oh,
There's electron transport.
oh.
I know. It's all gonna be fine.
Look at this. Oh, no.
It's not gonna be fine.
Oh,
Are you able to put out the answer sheets of the outline follow-up thing?
That would be great.
we can. I don't have time to work on that. I'll find
Whenever.
some time.
Just before the
Whenever,
next test.
just before the next test. Okay. All right. The whole point of respiration is to take all of this reducing power and to funnel it into moving around electrons to make ATP. So the Uber doesn't just keep driving around, okay? You want to get out of the Uber somewhere.
Uber driver.
Oh my lordy, okay? Every one of those NADH and FADH2 molecules are coming here to do oxidative phosphorylation and make ATP, okay? What we are doing, those electrons represent energy. And as we move the electrons, we're going to generate what's called a proton motive force. to ultimately help us make ATP, okay? This is a diagram you want to understand, okay? So here's what happens. NADH, remember, is our Uber, okay? It's carrying, I know, I'm going to keep saying it now just to drive you nuts, okay? NADH is carrying our electrons. The electrons are what we're going to use the electrons, we're going to use the protons as well, okay? So the NADH gets converted into NAD. In this process, the electron is going to go through the membrane. When the electron comes out of the membrane, it joins with the electron acceptor.
Wait, so all of this happened in the.
This is happening in the membrane.
Where is it?
Where do you think this other stuff happens?
Inside.
In the cytoplasm.
Oh.
Remember, prokaryotes have none of those membrane-bound organelles. That's all happening in the cytoplasm. This is happening in the membrane. So your Uber is going from the cytoplasm, wherever it's at, wherever in the cytoplasm it's happening, up to the awesome cell membrane. Through the highway to the cell membrane. When it gets here, it goes electrons, protons, you're all kicked out, okay? The electrons go through the membrane and they come out to join with, in this case, it's joining with oxygen. So what kind of respiration are we doing?
Aerobic.
Aerobic.
Woo.
Oh, it's on the side. Okay. Aerobic respiration. The protons, remember H. H, one proton, one electron. The electrons are going through the membrane. The protons are being pumped through these awesome things outside of the membrane. Look, NADH dehydrogenase. What type of molecule is this?
Awful.
It's an enzyme, which means it's made of
Protein.
Protein. Awesome. Okay, so we're embedded in the membrane. Okay? The protons build up outside here. It makes what's called a proton motive force. All this means is there are tons of protons next to one another. And they want to go from high concentration to low concentration. Okay? Have you ever been in an elevator with like 20 people?
Oh yeah.
What do you want to do?
Jump up and down.
You want to leave. You want to get out of there. So what happens here is that all these protons build up upside here, and then they're like, wait a minute, I don't want to stay here. I want to go back in where there's lots more space for me to sit. Okay? So the protons come back in through this molecule, ATP synthase, yes, also an enzyme, also made of protein. And when it does this, we make ATP. Okay? So, NADH, FADH2 is going to do the same thing. You release electrons and protons. The electrons go through the membrane and they come out and they join with the final electron acceptor. The protons go from inside to outside. Okay? This is the cell membrane. What's way out here?
Wall.
Out. The cell wall. So the cell wall is outside here. There's just a little gap. Okay? So the protons build up between the cell membrane and the cell wall. When they come back in, you make lots of ATP. Okay? Right? What's coming to the electron transport chain? What are we coming in with?
NADH.
No.
NADH
Oh.
ADH. I knew that.
Why do you say?
and FADH2. How many?
Two.
Nope.
4.
Nope.
6 NADH.
2 plus 2 makes 4 plus 6.
10.
We've got 10 NADH coming here.
Still nice.
How much FADH2? Just
Four.
the 2. Okay? Oops, starting material.
No way, it says FADH2. Isn't there 2? Is it 4?
No, there's 2 hydrogen in the molecule itself.
Okay, no, never mind.
Oh my god.
10 NADH, 2 FADH2. leaving here. This is going to become just NAD plus. And how many of those are we going to have?
10.
Awesome. This is going to become FAD plus. We're going to have 2 of those. But what are we driving with this
ATP.
process? We're driving ATP production. If it's aerobic respiration, how many ATP are we making? Our total number before was 38. We've made 4 already. So we
34.
gotta make 34 ATP. We'll spend some time next week. We're totally not gonna finish the slides today, I promise. It was intense. The goal is to take that reducing power and to use it to make ATP in the end. For every, so this is, you're not memorizing these numbers here, okay, but I will help you make sense of this table, okay, this figure. For every NADH that approaches the membrane, you're actually going to pump out 10 protons, okay? When those 10 protons come back in, you're actually going to have the ability to make 3 ATP out of that process. So one of these little Ubers is going to have us make lots of ATP in the end, okay? The whole point of that enzyme, ATP synthase, is to take the energy from the proton motive force, which is just all those protons on the outside, and allow the protons to come back into the cell and make ATP. For every 3 protons that come back in, 1 ATP is formed. If there are 10 protons pumped out per NADH, that makes 3 molecules of ATP. I know, you're like, wait a minute, 3 times 3 is 9, you have an extra proton
Yeah, I
left.
love it.
It all evens out in the end, okay? We make 3 molecules of ATP. 6 protons are pumped out per FADH2, which means 1 FADH2 is going to make 2 molecules of ATP. So the more efficient this process is, the extra protons just hang out there and they keep coming in groups of 3, okay? We're making those 34 ATP because we're bringing all of that NADH and all of that FADH2 to this membrane. Back from our original figure, we said there was 4 ATP created through substrate level phosphorylation because our glycolysis is netting us 2 ATP. Our TCA cycle is getting us 2 ATP as well. We're doing aerobic respiration. We're making 34 ATP from all of that NADH that's coming back in. So our theoretical maximum is 38. If you are ever asked, whether it's the next exam or the final exam, how much total ATP is made in aerobic respiration in a prokaryote, the number is 38. If you find any figures at all that are talking about this in a eukaryote, you will see 36 as the number. Because you actually lose 2 ATP with that coming across the mitochondrial membrane. So don't get bogged down if you find a figure and it says it's 38 or 36.
Okay.
Okay?
Okay.
If you know this for aerobic respiration, you also know it for anaerobic respiration. It's the exact same thing. The only difference is that final electron acceptor. That final electron acceptor is what makes a slightly inefficient process and a different amount of ATP. Okay?
Because they're not taking in those protons.
Because they're not taking in those, they don't have, right. They're using something else, and they're doing something else with the electrons. For example, E. coli in your gut. In your gut, that E. coli is doing anaerobic respiration. It actually uses a different carrier. It uses the quinone carrier. And as a result of that product, it actually produces vitamin K. E. coli is good, normal flora in your gut and is actually producing a vitamin that you absorb and you use. It's giving it off as a byproduct of its respiration. Okay? It's good stuff. We're going to, yeah. this is not that complicated, but let's just stop here for today. Don't go anywhere.
Can I take a picture of the board again?
Yes.